Hello there, welcome to this tutorial on A-level respiration. This is part 2 of the video. These are the learning objectives we covered in part 1. If you haven't watched it already, I highly recommend you do so you understand the flow. The link is in the description below. So these are the learning objectives we'll cover in this video today. So we'll explain in detail what happens during the Krebs cycle and what happens during oxidative phosphorylation, understand what is chemiosmosis, and also what other molecules can be used in the Krebs cycle. So let's get started. Okay, let's start with the Krebs cycle. It's ultimately needed to make many reduced coenzymes and ATP. All you need to know is that for one pyruvate molecule you make one ATP, one FADH2, also known as reduced FAD, and 3 NADH, again also known as reduced NAD. The FADH2 is another coenzyme like NADH, and the specificity of its function is beyond the scope of A-level biology. A thing to note is that the ATP made during this cycle is due to substrate level phosphorylation. Here, a compound directly gives phosphate to make ATP. I have not shown it in this diagram as you don't need to know what the compound is, but you do need to know the name of the process, that is substrate level phosphorylation. And I highly recommend that you redraw this diagram from memory. So let's come on to the final process in aerobic respiration. This is oxidative phosphorylation. It's a complicated process to understand, but let's take it step by step. Firstly, the process ultimately involves making ATP from oxidation reactions, i.e. through the loss of electrons. The process occurs in the electron transport chain, which is attached in the inner membrane of the mitochondria. The electron transport chain consists of electron carriers, which are proteins used to pump protons into the intermembrane space. Let's have a closer look at the steps involved in oxidative phosphorylation. Firstly, the reduced FAD and NAD are oxidized, releasing protons and electrons. Next, the electrons move from one carrier to another, releasing energy. This energy is used to pump a proton to the intermembrane space. Now let's look into this diagram. There are some details you don't need to know, such as the name of the electron carriers. However, it's good to understand that as you go along the electron transport chain, the electron released moves from a state of high energy to a state of low energy. You will learn about free energy, also known as Gibbs energy, in A-level chemistry. But if you're not, unfortunately you don't have to know this for A-level biology. Okay, next. The protons that have moved into the intermembrane space create an electrochemical gradient, as there's a difference in proton concentration between this space and the matrix. Note that in A-level we use the term electrochemical when we refer to ions. Next, the protons move down this electrochemical gradient via a transmembrane enzyme called ATP synthase. This enzyme catalyzes the formation of ATP from ADP and inorganic phosphate. This way of making ATP is known as chemiosmosis, which is defined as the movement of ions through a partially permeable membrane down a concentration gradient. Note the similarity of the definition to osmosis. Finally, once the ATP is made, the proton that moved through the ATP synthase oxidizes oxygen, which also gains the electron from the electron transport chain. Together, they form water. Okay, a thing to know at A-level is that other molecules can be used as substrates for respiration, such as fats and proteins. This is why if you don't eat carbohydrates and the so-called keto diet works, because if you don't burn the carbohydrates, you start burning fat and the proteins. The fat can be used to make acetyl-CoA by a process called beta oxidation, and the same can be done with proteins via a process called transamination. However, don't delve too deeply as you don't need to know the details. The acetyl-CoA made goes back into the Krebs cycle, also known by its older name as the citric acid cycle, and the process starts once again. So here's the recap of what we covered in this tutorial. Use them as questions to consolidate your understanding. Thank you for watching, and I'll see you in the next one.